Small intestines are a tubular organ that goes from the pyloric sphincter at the stomach to the beginning of the large intestine, the ileocecal valve. It fills up most of our abdominal cavity. It gets chyme from the stomach, along liver and pancreatic secretions in the duodenum, and it completes the digestion of nutrients within the chyme. In fact, most of the digestion that occurs will happen in the small intestines, and it is also important because this is where we absorb the products of digestion. Most of the absorption comes in the small intestines. We transport the remaining residue into the large intestines, which we'll talk about in the next section, and get rid of it. Small intestines are divided into three sections: the duodenum, the jejunum, and the ileum. The duodenum is the shortest, most fixed portion of the small intestines. The middle portion, the jejunum, is thicker and more active than the ileum. That's where a lot of digestion and absorption occurs. The distal portion, the ileum, contains the Peyer's patches, which are part of that mucosal associated lymph tissue. Jejunum and ileum are suspended by a double layered fold of peritoneum called the mesentery, and the mesentery is important because it's the blood and lymphatic vessels and nerves that go to the walls of the intestine. A double fold of the peritoneum called the greater omentum drapes down from the stomach over the large intestines and folds of the small intestines. So here's an image of that, the mesentery there connecting, and then a greater omentum and lesser omentum are part of that structure as well. Here we see the greater omentum covering the front. Teeny projections from the mucosa are called the intestinal villi. They are there to increase surface area so that we can get more absorption of digestive products. And each one of those villus is made up of simple columnar epithelium with a core of connective tissue, blood vessels, and a special lymphatic capillary called a lacteal and nerve fibers. Those lacteals are going to be important for absorbing fats from our diet. The free surfaces of those epithelial cells also contain microvilli, which further increase the surface area available for absorption. There are intestinal glands and crypts located between the villi. The Lieberkühn, crypts of Lieberkühn, those are an archaic name. We should be using anatomical terms that are more descriptive than that, but they are there. The Placiae circulae are circular folds of mucosa, and again, increasing surface area. This is where we absorb, and when you absorb, if you want maximum absorption, you need maximum surface area. And there are many modifications to help with that. So we've got a villus, and then if we magnify that villus, we can see almost make out what I described to you as the brush border. Those are the microvilli on the surface. And then they are shown here. And this is an electron micrograph. You can see it more magnified, and it looks really like a brush, and that's called the brush border of, again, increasing surface area. The small intestine secretes mucus from goblet cells. They are called Brunner's gland in the small intestines, and they have an alkaline mucus to balance the acidic materials coming out of the stomach. The intestinal glands secrete a watery fluid that picks up digestive products and transports them to the villi. It doesn't have digestive enzymes on its own. Enzymes in the membrane of the microvilli, though, include peptidases, some sucrase, maltase, lactase, and lipases. The peptidases break down peptides into amino acids or into short polypeptides, 3 to 4 amino acids. Sucrase breaks down sucrose, maltase, maltose, lactase, lactose. And those are disaccharides. Those are our dietary sugars. It makes them into monosaccharides, principally glucose. Lipases break down fats into fatty acid and glycerol. So there's a great deal of enzymes involved, some of them from the salivary glands, some of them from the stomach, some from the intestines, yes, and many of them, most of them, from pancreatic sources. Regulation of small intestine secretion occurs by mucus secretion in response to mechanical stimulation or irritants such as gastric juice, which is quite acidic. Contact with chyme will stimulate the goblet cells to secrete mucus and intestinal glands to secrete their watery fluid. Distension of the intestinal wall will activate nerve plexuses in the wall of the small intestine. Distension also stimulates parasympathetic reflexes, triggering the release of intestinal enzymes. So again, the villi increase the surface area for absorption. The small intestine is the most important of the absorbing organs in the alimentary canal. Small intestine absorption is so effective that very little absorbable material ever reaches the far end, the ileum end of the small intestine. For carbohydrate digestion and absorption, salivary and pancreatic amylases break starches and glycogen into disaccharides. Intestinal enzymes then break the disaccharides into monosaccharides. Those monosaccharides are absorbed by facilitated diffusion and active transport into the blood vessels of the villi, or in the villi. Facilitated diffusion are receptors that will allow sugars through. Active transport requires the pumping of those sugars. Protein. Pepsin breaks down proteins into polypeptides. Pancreatic proteases such as trypsin and chymotrypsin break down proteins into polypeptides into even smaller peptides. Intestinal peptidases break the peptides into amino acids, and amino acids are absorbed by active transport into the blood vessels of the villi. Fats. Fats are emulsified by the bile salts. Fats are digested mainly by enzymes from the pancreas and small intestines. They are digested into glycerol and fatty acids. The fatty acids and glycerol are absorbed by a process involving several steps. They are absorbed into the blood or into lymphatic capillaries called the lacteals. When they get into the I won't skip that, but let me go back. Okay, so you should go over this figure. Fatty acids result from fat digestion, and they enter the epithelial cells. Inside the epithelial cells, fatty acids are used to synthesize and synthesized in the endoplasmic reticulum. Those fats collect in clusters that are encased in proteins and form a structure called a chylomicron. That chylomicron leaves the cell, goes into the lymph lacteal, and in the lymph it is carried back through the lymph system to the subclavian vein and back into the circulation at that point. All right, so here's an overview of the mechanism and the means of circulation for a number of nutrients. You should review that. Within the small intestine, we get peristalsis and segmentation. Talked a lot about those. I felt like I did on Monday. Peristalsis moves things forward. Segmentation is breaking up the areas of that. And the images I showed you on Monday are quite descriptive of what happens there. But the main point is that peristalsis moves things forward. Segmentation mixes those things as they are moving forward. There are parasympathetic impulses that stimulate mixing and peristaltic movements. The sympathetic ones tend to inhibit them. And the small intestines becomes distended or irritated. There is a strong peristaltic rush that could sweep the contents quickly into the large intestines, and that can result in things like diarrhea. The small intestines join the large intestines, which the first part is called the cecum, at the ileocecal sphincter or ileocecal valve, and that's going to help regulate flow on the other end of the small intestines as well. All right, that wraps up the small intestines. Next, the large.